We are going to begin with today's webinar on glycosaminoglycan interactions uh, with virus envelope proteins. These webinars are being conducted by IDP project of GADVASU. For today's webinar, we have a very eminent speaker with us, Dr. Ritesh Tandon. He is Associate Professor in University of Mississippi, USA. Dr. Tandon has done his PhD from University of Georgia and has significant experience in virology and immunology. He has made important discoveries related to virus and host protein interactions of cytomegaloviruses. He has also acquired funding for research projects from American Heart Association, USDA, NASA, and several other sources. So now I request Dr. Tandon to start with the webinar, please.
Okay, thank you, Gurpreet, very much for the kind introduction. So my name is Ritesh, and I do have veterinary background. I got my BVSc from Pantnagar, GB Pant University back in 2000. And I did some research at IBRI, Izzatnagar as well. And after that, I started my PhD program in veterinary parasitology. And I was working on horse parasites, small strongyles at that time, and got very interested in molecular biology. And the problem with parasites was it was a very difficult system to manipulate genetically. We just had to necropsy a lot of horses and get the parasites from intestine to do anything genetically. So I moved into the virus system because, as you all know, viruses are very, very small, very small genomes and very easy to manipulate. So it's very easy to study a phenotype. If you have a virus, there are genetic systems available for the viruses. So you can take a virus, you can modify any way you want. You can make point mutations or you can delete genes, and you can study what's going on with those viral functions. So my career mostly has been, after my PhD, mostly focusing on herpes viruses. And with herpes viruses, and I've been involved mostly in human viruses after that time. Herpes viruses are very important in veterinary field. And I think there are students there, they should be reading about Marek's disease virus in chickens and ILT and pseudorabies. So very, very important viruses in the veterinary world. But in human, there are three categories of viruses. They're classified as alpha, betas, and gammas. And alpha viruses such as HSV1, HSV2, and VZV, they're responsible for the herpes disease, as we know, the oral and genital herpes. And VZV is the virus that causes chickenpox in early childhood, and also shingles later in the life. The virus I have focused most on is cytomegalovirus. Not a lot of people know about cytomegalovirus, but it's very important, and I will talk to you about this in a bit. Then this beta group also includes HHV6 and 7. And then gamma group involves viruses that cause cancer. So these are oncogenic herpes viruses. And all of these viruses look very similar. They have a double-stranded DNA genome inside. You can see this is just a cartoon. And it is surrounded by a capsid. And this is further surrounded by a layer of tegument. And it is surrounded by envelope. So these are, as far as viruses go, herpes viruses are pretty complex. They have huge genomes that can range up to 250 kilobases, which is very large for a virus, considering you can have a virus as small as 1 or 2 kb. And they all are enveloped. And envelope that's outside, it's a lipid envelope. It has virus glycoproteins embedded, and it plays important roles, not only in virus entry, but in pathogenesis as well. So CMV is important, and it's a virus that pretty much everyone has. So it's one of those latent viruses, and we acquire it during early stages of life. And there is surveillance data to show that 80 to 100% of the people in the world already have it. This stays dormant, so it does not cause a lot of issues in people who have an intact immune system. So if you get immunocompromised, then it starts to create problem. And the problem that it causes range from encephalitis, retinitis, pneumonia, gastroenteritis, and most importantly, in a pregnant woman, it can cross the fetal placental barrier and can go into the fetus and cause congenital developmental abnormalities. And it's known, and it's a secondary factor in a few cancers and vascular diseases such as atherosclerosis. So it can lead to cardiovascular issues as well. But what's known about the prevalence, and this is data from US CDC. I don't have data from India, but in US, congenital CMV causes more cases of developmental abnormalities compared to Down syndrome or fetal alcohol syndrome. So we know a lot about Down syndrome. We know that no one should be drinking alcohol during pregnancy because it can cause developmental disorder, but we know very little about CMV. But this is the most common cause of congenital developmental disorders. So there is no vaccine for this. I know we have been trying to develop vaccine for a lot of viruses. There is no successful vaccine yet for CMV. There are few drugs available. None of them are approved in pregnant women, and there's some available in the organ transplant settings where you have to put people on immunosuppressive therapy. But these are very toxic and have side effects. So one of our goals has been to develop mainly antivirals. I've not been working on vaccines as such. I've been working on some viral proteins, but my area has been trying to develop antiviral drugs. So the other virus I'm going to talk to you about today is SARS-CoV-2, and it does not really need an introduction. That's why I don't have any introductory slides here. Pretty much everyone knows about SARS-CoV-2. It's just amazing and unprecedented. more than 86 million cases globally, and almost 2 million people have already died from this virus. So now we have a vaccine that seems to be working well. But now there are other strains emerging in UK and in South Africa that are causing issues. And we don't really know if the vaccine is going to be effective against all these newly emerging viruses. So it's also important to study antivirals and viral properties of SARS-CoV-2. So these are the two viruses I will be talking to you about. So the direction I want to go today, and I know I have done a lot of work on virus assembly and maturation, egress. This is more related to virus entry. And the field tends to focus on protein receptors. So we know ACE2 is a known receptor for SARS-CoV-2, and there are known receptors for CMV and herpes viruses, but what's been largely ignored is glycans or sugars on the cell surface. So our cells, all cells have sugars on the surface, and they form a layer on top of the cells that can be important for virus to attach and enter the cells. And some of the work that we have done and Jeff Esco's group has done at UCSD has shown that there are patterns of these sugars. So these are just not inert molecules. These molecules have a specific interactions with virus glycoproteins, and this interaction can determine how and if virus enters the cell or not. So the background to say is that pattern of glycans or sugars on the cell surface can cause pathogens such as viruses as well as bacteria to enter certain cell types, and that will determine which kind of tissue and cells the virus would infect. And a particular category of these sugars known as heparan sulfate glycosaminoglycans, and I will just say GAG here. These are complex polysaccharides that contain amino group, and these are present on almost all cell types. And they can function as primary cellular receptor for mostly enveloped viruses. So not going into the naked viruses at this time that have a slightly different mode of entry. But enveloped viruses such as SARS-CoV-2 or CMV and even dengue virus, they all have a lipid envelope. And these sugars may have an important role in interacting with this envelope glycoproteins. So this is a cartoon of how virus entry occurs in herpes viruses. And if you see on the right, these are virus particles in red, and this is the cell layer. So what's known as virus particles, they kind of scan the cell surface to find an optimal place to enter the cells. And if you see this depression, this is formed by uh, an actual event that occurs between the virus and cell surface, known as fusion. So unplugged viruses can either be endocytosed directly, or more often they go a fusion at the cell surface. So if you see on the left, there are different glycoproteins in herpes viruses, and there is a glycoprotein named glycoprotein C in HSV1 that is known to bind to these heparan sulfate sugars on the cell. Now, this is a primary interaction, and this interaction tends to concentrate the virus on the cell surface. So viruses find a spot to attach to. And then the interaction is further mediated by secondary and tertiary interactions, such as glycoprotein D and HVEM interaction shown here on the left in binding. And finally, this leads to membrane fusion. And in the fusion, the virus envelope stays outside, and it fuses with the cell membrane here. And virus envelope literally does not enter the cell. It's only the capsid that enters the cell. And most DNA viruses, with an exception of pox, would actually go and dock to the nuclear pore in the nucleus of the cell and deposit their DNA into the nucleus. And that's where virus replication and protein expression and all those things happen. So this is a very simple diagram of virus entry in herpes viruses. SARS-CoV-2 is RNA virus, so these things don't happen in SARS-CoV-2. The replication is in cytoplasm. So what we don't know is how herpes viruses, or a matter of fact, other viruses interact with cell surface GAGs. And if there is specific GAG modification, such as sulfation, that would govern this binding. So our hypothesis in this project is a specifically sulfated heparan sulfate on cell surface mediate CMV entry, and thus the entry can be blocked using a specific heparan sulfate. So these are different kind of glycosaminoglycans on the cell surface, mainly 6 different kind. So you see hyaluronic acid, which is not sulfated. Then you have heparin and heparan sulfate, which are very similar molecules, but different. They're structurally very similar, but they have differences in the number of units and number of sulfations and those things. Then there is dermatan sulfate, chondroitin sulfate, and keratin sulfate. And they're all made of these basic sugar units. If you go into the carbohydrate chemistry of these, so there is N-acetyl glucosamine and glucuronic acid that are the main subunits in a lot of these. So I won't go too much into the details of the structure, but just wanted to point out that these sugars, these are disaccharides, and they can be sulfated. So they can be sulfated at different positions of the carbon. And these sulfations, such as thiosulfation shown here, can be important in determination of their structure. So first experiment we did a few years ago when we were starting to look into this interaction is we designed a glyco array. And what this array is, it's a glass slide basically, and you can use a robot to spot different glycosaminoglycans on a chip. So we spotted these hundreds of different glycosaminoglycans. And they were all done in 8 replicates, and they had negative and positive controls. And these are the different glycosaminoglycans that we were using in array 1. And I don't want to go into too much detail of their structure, but what's important is we used a few of the hyaluronic acid GAGs, some heparin sulfate, some chondroitin sulfate, and dermatan sulfate GAGs. So we used different types of GAGs because we wanted to see what kind of GAGs would bind virus. And this is a human cytomegalovirus, and this is how the data output from this glycoarray or microarray looks like. So you have a fluorescent virus, and you pass it over this glycoarray, and everything that binds would light up and would give you a signal. So this would be positive control here, and then you have some sugars that are binding. So what are these sugars that are binding? If you look at the data that comes out, and it's plotted as a relative fluorescence unit on the y-axis here, you have most of the binding that we see. So an interesting thing to note, and this is purified extracellular CMV that's fully enveloped. It does not bind to hyaluronic acid at all. Now it does bind to heparin, and as you increase the branch length, so this is heparin going from a lower branch length to a higher branch length, and this is almost a DP. DP is a degree of polymorphism. So you have a branch length of 30, and this is the strongest binder in this assay. And now there is very little or no binding to chondroitin sulfate, and some binding to chondroitin sulfate D when it's very highly branched, and some binding to dermatan sulfate, and these are controls. So what comes out of this is heparin or heparin sulfate are the molecules that is strongly bind to CMV on a glycoarray. Now we can further refine this binding with heparin sulfate. So this is another glycoarray that we used. And the idea here was to use unsulfated heparin and see if they bind to virus or not. And also different kind of sulfation. So we have aminosulfated and amino-6-sulfated and mixed sulfated heparin sulfate. And this is the number of sugar residues. So it tells you what kind of degree of polymorphism they had. And this is sulfate groups per disaccharide. So this tells you how much sulfation they had. So this is the red shows you here the molecules that had highest number of sugar residues. So these are the longest molecules. And the degree of sulfation goes up here. So we wanted to assess if it's the degree of sulfation or is the number of residues or is it both. And if you look at the data, most strong binding we saw was with these molecules, 14, 15, 16, that had a moderate degree of sulfation, but the highest chain length. So if you go back, you see these are the molecules that had a chain length of 9 and average sulfation of about 1.6. So what that tells you is that both degree of sulfation and chain length are important, but it's not only sulfation. So if you keep on increasing, this is a very high sulfated molecules, but the chain length goes down. So having a good balance of sulfation and degree of polymorphism is important for binding of CMV on this glycoarray. So with that information, we went a little bit into drug design and some medicinal chemistry. So we got these anticoagulant medications. These are low molecular weight heparins, known as enoxaparin and Erixtra. These are FDA approved and they're used as an anticoagulant in clinic. We collaborated with a chemist, Josh Sharp, and he purifies these sugars. And what he did was he did a fractional purification of enoxaparin. And he got these sugar molecules with different chain lengths. So you can have a chain length of 4 to 20. And we use these sugars to see if any of these sugars would block virus binding. And this is the data from titers. So in virology, titers really mean how much virus you're getting. So you treat the cells with these sugars and you see if you get any virus out or not. And if you get virus out, it means that there is no block in entry. If you don't get virus out, it means there is a block in virus entry. So we do the titers on cells that we treated with these different sugars that we had. So if you look at the titers on the y-axis here, and these are different molecules that we were using. PIHSS is the heparin from porcine intestine. So it's just the normal heparin that we get from natural sources such as pig intestine. And this is commercially available, and it is very effective in reducing the titers to, and this is on log scale here, so almost 100-fold reduction in virus titer when you're using heparin treatment. And these other molecules, especially the derived molecules, if you have a very high chain length, a degree of polymorphism 20, you can get a pretty good reduction in virus titers, but not when the chain length is small. So this again proves that a degree of polymorphism or chain length of sugars is important in binding to the virus or reducing viral titers. And this is plotted as titer versus degree of polymorphism. So as the degree of polymorphism or polymerization increases, you get less and less titers. And other thing we did was, whenever we are looking for an antiviral, we want to make sure it's not toxic to the cells itself. So the cells that are infected or uninfected, they're treated with these sugars. And this is cell viability plotted here. And you can see when cells are infected, they would naturally die. So this is done in about 5 days. So you would see about 70% cells die when they're infected, and they don't when they are not infected and treated with these molecules. So these molecules themselves are not toxic to the cells. And when cells are infected, these molecules can actually prevent virus-induced cell death. So as you see here, virus naturally would kill about 70% of the cells in about 5 days at a very high multiplicity of infection. And when you treat with heparin, you can have those cells recover and not die, because heparin is actually blocking virus infection. And that's true with the increasing degree of polymorphism or branch length for these molecules that we were trying. So with these, another thing we wanted to do is look in the actual cell system. So we looked into glycoarrays, we looked into different mimics or different sugars, synthesized sugars. We wanted to go ahead and see if we can block virus infection in cells that are mutant. So these are different mutants that our collaborator, Dr. Wang, made. And these are mouse cells. They are derived from different mice, and they are knocked out for these different enzymes. So these are the enzymes that would actually sulfate the heparin. So, and there are different kinds of enzymes. So HS3 is a 3-O-sulfation, HS6 is 6-O-sulfation, and so on. So all these are somehow or somewhere implicated in the heparin sulfation pathway. And they've been mutated with CRISPR-Cas9 or Crelox-based system. So when we looked at some of these mutants, we saw that if you infect cells with CMV and the cells are pre-treated with these, or cells are not pre-treated but are actually knocked out for these different enzymes, you have a reduction in viral titers. So this is the 3-sulfation that's very important for virus binding. And if you knock out the 3-sulfation, the virus titer goes down. And this is shown at low multiplicity as well as high multiplicity here. So at 6 days post-infection, you have almost 200-fold reduction in viral titers when you treat these mutant cells with the virus compared to wild type. And now the fourth thing we had, or the fourth tool we had these peptides that were raised or designed against heparin sulfate. So this is a peptide that was raised against wild type, and this is a peptide that was designed against 3-O-sulfated heparin sulfate. And when you treat cells with these peptides, you also see reduction. So this is the fourth point, or the fourth type of experiment that proves the same point, that heparin sulfate are important in virus binding or infection. So to conclude this part, I would say that CMV binding to cell surface glycans is dependent on branch length and sulfation of HS. Now, when SARS-CoV-2 came along earlier last year, we were tasked to design some antivirals for SARS-CoV-2 and design some assay to really do some antibody titers and determine what kind of entry factors are involved in SARS-CoV-2 and receptors. So not going too much into details, because we have heard so much about SARS-CoV-2. So SARS-CoV-2 is a coronavirus. It's a positive strand, single-stranded genome that's surrounded by an envelope. And this envelope has this glycoprotein, known as spike glycoprotein, that's present in the trimer form. And this is the glycoprotein that interacts with the cell, mainly using ACE2 receptors. That's been very well established. And the spike protein has 2 fragments. It cleaves into S2 and S1, and S1 fragment binds to ACE2, whereas S2 is involved in fusion of cell and host cell and virus membrane. So neurophilin 1 is also involved, and we've been looking into neuropilin-1 because that's also involved in CMV infection. But these are protein receptors. We are more interested in the sugar receptors at this point. So what we did was we didn't want to deal with the actual virus because that requires a high level of biosafety, and we could just pseudotype these particles. So what is pseudotyping? Pseudotyping is fairly simple. We use it on a regular basis in a virology lab. So you take the envelope glycoprotein from one virus and you put it on another virus that's not hazardous. It's done very frequently in viruses such as Ebola and Marburg, because they're very difficult to work with and require high level of biosafety. So SARS-CoV-2 require a BSL-3 type facility that's difficult to get in, and involves a lot of regulation in those things. So we took the spike glycoprotein from SARS-CoV-2 and we pseudotyped it or put it on the backbone of a lentivirus, which is HIV, but is defective in replication. So we made this virus, and this is the schematic of how we made it. So you take some mammalian cells such as 293Ts and you put in all the components of lentivirus along with the glycoprotein from SARS-CoV-2, and you put a reporter such as a green fluorescence expressing lentiviral backbone. And the particles you get out of this are virus particles that have a body of HIV, non-replicating HIV, but the surface of a coronavirus, which looks exactly like coronavirus to the cells, because when it enters, these are the surface glycoproteins that interact with the cells. And if you have cells that are expressing ACE2, you can use this virus to infect these cells. And the cells that are infected, they turn green. So that's our output. That's how we know that virus is infecting the cells, and the cells turn green. And this is data from another group just showing that a pseudovirus is as effective in binding to antibodies as a live virus. So if you look at the red particular antibody P2C1F11, the binding kinetics of this antibody to the live virus is very similar to the binding kinetics of the pseudovirus. So this is proof of principle that pseudovirus is not really any different than a live virus when it comes to a virus binding to antibodies or to binding to cells. And this is our transduction, which means it shows you that we can get this reporter expression when we pseudotype our lentivirus or our retrovirus with a control vesicular stomatitis virus G glycoprotein or CoV2 S glycoprotein. And PLV is much more effective. That's why I use the lentivirus backbone for this. And this is some of our initial data that showed that if you have cells, and these are 293Ts, and they have been transfected with the ACE2, human ACE2 protein, plasmid, expressing plasmid. And you can see the protein is expressed in very high quantities here in these cells. There is also a little bit expression in 293Ts that were not transfected with this plasmid. And the degree of transduction actually depends on if you have ACE2 or not. So having very high ACE2 actually gives you very high transduced GFP cells there. Now, how do we use this assay? So this assay was used initially to study neutralization responses in patient samples. And these are two patients I'm showing as an example. And the patient N304 had a RT-PCR that was positive, N308 had RT-PCR negative. And they did have, N304 had very high level of IgM, IgG responses. N308 was practically negative. And you can see here when you use this serum, patient serum in this assay, we see a very high degree of inhibition using N304 because he had higher antibodies. And this shows that the pseudovirus assay replicates what happens to the virus in patient serum. Now, so the idea here that I was alluding to earlier is that SARS-CoV-2 would bind to heparan sulfate on the cell surface. And then this binding would stimulate binding to ACE2. And this ACE2 would lead to either endocytosis or fusion of the virus and host membranes. Now we looked at different sugars in this respect, and obviously heparin was a good candidate because we already had data on heparin from CMV studies. And we looked at a few natural sugars from different marine organisms. Um, and our chemist friends, they actually desulfated them or selectively desulfated to try to figure out what kind of sugar structures are important in binding. And these are all characterized by NMR. Now, this is the pseudovirus assay that we use for patient antibody screening, and the same assay can be utilized for studying virus entry. UFH here is ultra-fractionated heparin, which is just a fancy name of heparin that we have purified. So UFH, you can see blocks. So this is chondroitin sulfate, this is UFH. So UFH at 3 different concentration would very efficiently block GFP transduction, which means it blocks SARS-CoV-2. Now, if you have the N desulfated UFH, that activity goes away. So now this proves that sulfation is important. So if you have sulfated heparin, it's very effective in reducing viral titers or reducing transduced cells. And if you take the sulfation away, the activity goes away. And now, if you take the sulfation away at the 6S position, not the amino terminal position, the activities is still maintained. Now, this is very important because heparin, we all know, is an anticoagulant, and you can't really just give heparin to everyone, especially people who are more prone to stroke or bleeding disorders. You can't give them heparin. Now, 6-desulfated heparin actually loses the anticoagulant activity. But as you can see here, it is very effective in inhibiting virus infection. So this could be a really good molecule that we can design or develop further as an antiviral because it's technically heparin, but it does not have an anticoagulant activity. Now we found a lot of other, especially these natural sulfated galactans that are effective. These are difficult to commercially scale up. So right now we are focusing mostly on heparin and 6-desulfated heparin. This is more into what kind of inhibition they have and looking at their IC50s, and you can see UFH or our purified heparin has a pretty low IC50, which means it's very good. So you don't need to attain a very high concentration in blood for this to be effective. And the desulfated, 6-desulfated heparin actually has even higher, even lower IC50, which means it's even more effective. The others, such as enoxaparin, had a milligram per liter IC50, which is not very promising. So you want your IC50 to be somewhere in micrograms per liter. So UFH and desulfated UFH are very good molecules. Now we also confirmed these findings with something called SPR, which is surface plasmon resonance. And this looks for competitive binding of heparin. So you put biotinylated heparin on a chip, and then you look for binding of the virus to that chip. And this is done as a measurement of kinetics, like how much time does it take to achieve the full binding and how much time it would take to achieve an equilibrium. So all these data would point to a very effective heparin to virus binding. And later on, we looked at different oligosaccharides, and overall, the bottom line here was heparin was still very effective compared to dermatan sulfate or different chondroitin sulfates. So we are still focusing on heparin because of this. And this is looking into a degree of polymorphism or branch length. And the same thing, the same result we found with CMV is true for SARS-CoV-2. So as you go increasing the chain length, so this is DP of 18, and it looks like it's most effective. So branch length and sulfation, again, have a very high impact on virus binding. And this is a design where we are looking into either hypersulfating heparin or getting a non-anticoagulant form of heparin by chemical synthesis. And if you look into trisulfated heparin and non-anticoagulant heparin, we get similar data. And IC50 for heparin seems to be around 0.1 to 5 micromolar, whereas the trisulfated and non-anticoagulant heparin a little higher. So in this assay, it looks like the heparin may be the most effective molecule. But this is the matter of bioavailability as well. So these are all cellular assays, and we have data from mice experiment that show that heparin is really effective. Now we did, just as a proof of principle, we wanted to do this in a live virus assay. So we, not in my group, but in our collaborator groups in New York, they got live SARS-CoV-2 and used Vero cells, which are monkey cells, and infected these cells with the COVID-2 and used a pretreatment of non-anticoagulant or coagulant heparin and a few different molecules that they had. So if you don't have any sugars, you see there's a lot of viral plaques here. This is no virus here, so this is how negative would look like. And if you look in heparin, the virus titer is much reduced compared to no polysaccharide. So it shows you that these molecules work in live virus settings as well. Now, to conclude all of this, I would say we, with the SARS-CoV-2 project, we were able to design antiviral serotype particles, and these are very useful to study neutralizing antibody responses, because you can have antibodies to SARS-CoV-2, but they may not be neutralizing, and that's been a big question in the vaccine field. Like, do you raise antibodies, and if you do, are these antibodies neutralizing? So these serotype particles are very useful in non-BSL-3 setting to study those responses, even after vaccine administration, to see if good antibodies are maintained over a period of time. And we use the same particles to screen for virus entry inhibitors and mainly focusing on heparin sulfate molecules. Now the poor activity of chondroitin sulfate, which is also sulfated, showed that it's not a question of just presenting negative charge on a linear polymer. It's more about the structure of the sugar that's important. And that's why these sugars are bona fide receptors for virus. And also when we did complete desulfation, specific and desulfation, it decreased the potency of both heparin and enoxaparin to inhibit SARS-CoV-2. So it showed that 6-0 desulfation had little effect on potency, which is very important because now you can have non-anticoagulant heparin to be used as a molecule in clinical settings. The pseudoparticles showed very tight binding to immobilized heparin. So that again shows that heparin could be a very good molecule to use as an antiviral. Now, we are still working on, this is an ongoing project. We have published some of it and it's still ongoing. So we're trying to further characterize this binding and trying to determine the optimal binding structures. We have some structural molecular biologists involved in this project, and they're looking into what kind of structures are really important and how can you design a molecule that's safe to administer as well as very potent. We have different, we have a clinical trial for the intranasal heparin going on right now. And there could be inhalation route or other routes that we can test to possibly avoid detrimental anticoagulation, because the very little heparin would be absorbed into blood from the nasal route. And that's one reason we've been looking into inhalant therapy. We're also working on long-term toxicity. We have some short-term toxicology pharmacokinetics data, but we are looking into long-term toxicology pharmacokinetics. So with that, I would like to conclude and just thank people here who've been involved in the project and put in a lot of effort into this. Especially at UMC, Dr. Marshall is running our clinical trials. Dr. Bates has been responsible for the antibody assays in the clinics. Then the students in the group, Deepa, Poonam, Martin, and Christian, who've been involved in these projects. And our chemist friends at University of Mississippi, Josh Sharp, Nicole Ashpol, Witter, Mahavir, and Ha. And also at RPI, people who did the SPR experiments, Dr. Linhart's group, and Fuming and Beva. Some of the funding came from internal resources for this, and we have a project with NASA trying to look for virus reactivation in its space. And NASA was interested in, when SARS-CoV-2 came along, they were interested in some of the data from SARS-CoV-2. So we diverted some of their funds to this project as well. So with that, I would like to just thank the department here and encourage you to apply for PhD programs. We have very good PhD programs that are fully funded by NIH for the duration of the study. And if you have any questions about the graduate programs here, you don't need a master's. All you need is a BVSC, and you can apply for the PhD program. And if you have any question, I will be happy to answer. But that's all I had for today. Thank you so much.
Thank you very much. It was a very good presentation.
Oh, thank you, Modit. Yeah, nice to see you.
Nice to see you also. I would like to ask Ms. There is some chat box, some question I'm compiling and looking at it. But one question particularly I'll be interested in is, why sulfation incited you to just focus entire research on, or do you had a previous clue whether this sulfation would really be effective in preventing attachment of your whatever viruses you are taking, either herpes virus or you are talking about SARS coronavirus? So why this sulfation only was the focus?
Yeah, so we looked at different things. So when you think about carbohydrates, you mainly think about the branch length. And sulfation is pretty much the main modification that happened to these carbohydrates. So there could be a few other things, but these are very simple molecules. These are not as complex as proteins. So they don't have like a ton of post-translational modifications. And sugars are not coded in our genes. So you can't really make a mutation in sugar, but you can make a mutation in enzymes that sulfate these sugars. So these enzymes are proteins. So all the mutations that we have done are actually in proteins that sulfate these sugars. So there seems to be a very, very vast category of genes dedicated to sulfating the sugars in the body. And that's a clue to why sulfation could be important. And we had some data earlier that showed that thiosulfation was really important for CMV to bind. And that's another reason we went to go into looking into sulfation patterns.
Two more questions which has come up. One fellow is very much interested that why we are going after vaccination, or we can also, the alternate strategy he's suggesting is that why aren't we focusing on antivirals? Because there are a lot of hues and aware of two vaccines which have been approved, and one vaccine has gone into controversy as per the media reports. So people, because everybody is watching nowadays very seriously at TV, so they are anxious. So the same fellow has asked, one student has asked this. Can you please elaborate?
Yes, so I'm a huge proponent of vaccines. Vaccines are very good. Think about it, smallpox and polio, diseases that have already been eradicated or almost eradicated. And vaccines work really well. And I guess it's more practical because you get one dose of the vaccine or maybe two, and you're protected for years or life. Now with antivirals, you pretty much have to take them every day. So with HIV, we don't have a vaccine, but it's been largely controlled with antivirals. But the problem with those antivirals is you have to take a combination pretty much every day, and people don't stick to that. So the reason we have not been able to eliminate HIV is people don't stick to those regimens, and those antivirals have a lot of side effects as well. So people don't like to take them. So it's more about convenience. You take one shot and you're protected for life. Everybody would go for that rather than having to take an antiviral every morning.
Okay, thank you very much. One more question is there. Why vaccine means, this was in WhatsApp also I have heard, and media also I have heard the same. One student has asked that why it has took so long to produce a vaccine. Still we don't have a vaccine for HIV. but so quickly, within a span of 9 months, we are a vaccine for a
Correct.
COVID-19 at least. So
Yeah.
they are very much interested in how this type of mystery has taken place in particular science world.
Yeah, that's a very good question. And I would say the reason is basic virology. So HIV is a very different virus, right? HIV establishes latency. It has a lot of genes that manipulate the human immune system. It just hides from the immune system. Now, coronaviruses, we have been lucky that they're very simple viruses. They don't mutate as fast. They have a proofreading enzyme. They're relatively simple. They don't establish latency. So the virology is much simpler in case of coronaviruses. And now the vaccine, so there are two things with vaccine. There is an antibody response. So you look for a good rise in antibody levels, both the early IgM type response and IgG, and maybe IgA type response. And if you get very high responses, and they're neutralizing. So neutralizing is very important. So with HIV, you can get high levels of IgM, but they're not neutralizing. Now with the coronaviruses, these antibodies are neutralizing. And that's what we have seen in our assays, and that's what people have reported. So the initial trials with vaccines, so vaccine design is fairly simple if you think about it. It's just one mRNA, right? Or one spike protein or inactivated virus. It's a very simple design. Now, does it work in clinics or not? It totally depends on what kind of antibody responses you get. And second is cell-mediated immunity. Are you getting good T cell responses? And we don't really know how important they are, but they probably are. And the third thing is memory. Do you get a good memory response, both B cell and T cell memory responses? And looks like we have been just so lucky with both the Pfizer and Moderna vaccine that we get very good B cell memory, at least. I don't know about T cells. They're very difficult to study, but you get a sustained immune response, you get good IgM levels, and these antibodies are neutralizing. With HIV, that's a very mysterious virus. So how do you deal with HIV reservoirs? So it's latent, it integrates into your genome. So once you get HIV, it's almost impossible to get rid of it. Coronaviruses are just hanging out there. They're not integrating into the genome. And they may have some residual RNA, after the infection is over, but they pretty much clear up. And you get a good immune response from natural infection as well. Very little data that's out there is people getting a second infection or not. Very, very rare cases they get a second infection. So I believe we have been just lucky with coronaviruses to say. I mean, we have not been lucky because it was very transmissible. It was very dangerous in the sense that mortality was really high in high age group. But overall, I mean, if you think about a scenario where Ebola virus becomes airborne, I mean, there is no cure to that. That would just destroy the whole planet.
Thank you, Ritesh. One more question is there. Then I found it very interesting. Will the immunity, like you have two vaccines, Moderna and Pfizer. Similarly, India is having another two vaccines. One is from Oxford University and another is from Bharat Biotech, Covaxin. So I don't know how much they will be stimulating, and you are knowing it, I don't know. But they are asking that whether if a mutant strain, which has been reported particularly from South Africa and as well as in UK, and you find that a lot of lockdowns are already imposed, particularly in UK, particularly that has been done. So will the vaccine protection imparted by the first vaccine, which you have been, I think you must have been vaccinated, will it be sufficient, or the protection will be sufficient to provide you protection for the mutant strain?
Yeah, and that's a very valid concern. And so far the data I have seen is the vaccine is effective, or at least the antibodies are neutralizing against the newly emerging strains that we have in hand. So especially the UK strain, there is data on UK strain. People have already done those mutations in the lab, and they showed that antibodies are still neutralizing against that. Now, the South African strain seems to be more dangerous because the antibody responses seem to be going down for that, or the neutralization responses seem to be going down. Now we don't really know yet because we don't have any data on the vaccine efficacy against these strains. But it's a reasonable concern to think about the newly emerging strains that could be coming up. And it's more because of the high transmission rate. If you could block the transmission in its footsteps, you won't be in this situation where you have, because if you think about mutations, they're very low frequency. There may be one in 100,000 or a million. So they don't propagate until the virus has a chance to replicate freely. So yeah, I cannot comment if the vaccine would be effective. The data that's out there shows that it would still be effective. So if you have an opportunity, go ahead and get the vaccine, because you are likely to be protective. And even if it's not going to be like totally ineffective. So like flu vaccine, yeah, you take a flu shot. It's almost never 100% effective, but it prevents the clinical disease, and that's what we are after. As long as it can avoid having to take people to ICU, this is still worth it.
Thank you, Dr. Ritesh. Thank you very much. Now I shifted to Dr. Gurpreet. Thank you very much.
Okay, thank you, Mudit.
Thank you, Dr. Ritesh. We have with us Dr. Deepthi. She's our head of the Department of Veterinary.
Yeah, I'm here. Yeah.
Hello, Dr. Deepthi.
Hello, sir. It is very nice and it was so informative. And I'm sure our children, they are, I'm really at their curiosity and how the questions they're asking. I think they have a good number of questions in their mind, but because of the paucity of time, I think I'm sorry for children, we won't be able to consider all your questions, but I'm really happy to see their response. And I'm so thankful, Dr. Tandon, and you spared your time for this webinar. And we are also thankful to our IDP team that they have really given us this opportunity to talk to you. And it was very nice lecture. Like you started with everything. I think the role of ACE2 receptors. Everybody's reading about these, but how nicely and how you have depicted everything, and this branch length and sulfation, and how how this small heparin, and it is just being like a miraculous drug, I would say, if something comes like that. So it's very nice lecture, sir. We are so thankful to you. Yeah.
Thank you, Dr. Deepthi. It was very nice interaction. I really enjoyed all the questions. I'm seeing all the questions from students. A lot of interest.
There are so many queries.
Yeah. Yeah, maybe I will try to get to those questions later. Maybe just email.
Questions, and if you can reply, we will send to individual students.
Great, thank you so much. It was very good experience for me as well. We always learn from presenting our work to others. We always get good questions that get us to think into new directions.
Yeah.
Thank you very much, Dr. Tandon. Thanks for the nice talk. Yeah, and it was very informative for students as well as for us. Thank you so much.
Okay, thank you very much. You have a nice Happy New Year.
Thank you. Same to you.
Okay, thank you. Happy New Year.
Okay, bye.
Okay, bye.
Okay, bye.
Okay,
Bye.
bye.
Thank you.